Medical technologist or medtech is all around you, and you probably don't even realize it. Medical technologists are the heroes behind the scenes who play a very important role in an individual's health and wellness. MedTech is part of your life from the day that you were conceived right to your maturity and until the highlights of your golden age. As soon as you are born, MedTech will test your blood as a part of the newborn screening. Later in life, when you get sick, MedTech routinely measures your health through samples like collecting blood, stool, or urine for a lab test to help the doctor diagnose what kind of disease you have contracted. You are introduced to even more MedTech lab procedures to monitor your vital stats and health status upon reaching the age of 40s and above through blood sugar monitors, lipid profile, and many more. These are the ways of the medical technologist which help keep you healthy on your journey through life. But what does a medical technologist do inside the laboratory? Here, we prepare a 3D display of the medical laboratory to provide a full virtual representation and conceptual experience for aspiring medical technologists and individuals.
Drop it.
The laboratory where specimens are received and sorted once they arrive or delivered. After organizing all the specimens, they are now prepared for processing. Samples from patients suspected of bacterial infections undergo several tests which occur at the bacteriology section. This is the culture bench where different human samples such as fluids, tissues, swabs, feces, urine, and blood are cultured in various media to check the causative agent of the patient's disease. In this area, microscopy and staining methods are done to look for any presence of bacteria under the microscope. As you can see, there are examples presented which demonstrate one of the many methods in bacteriology, which is a biochemical test, specifically the TSI test. The left tube indicates an acid slant and acid butt, while the tube on the right indicates an alkaline slant and alkaline butt. The incubator maintains an optimal temperature, humidity, and other conditions such as the carbon dioxide and oxygen content of the atmosphere inside. Some blood specimens during analysis are cultured in enrichment broths, which are put inside the incubator for approximately 18 to
48 hours later,
depending on what organism is suspected. Inside the bacteriology section, antibiotic susceptibility tests are also performed to diagnose significant organisms and determine the toxicity levels of the antibiotic that will determine the resistance and sensitivity of the organism. Laboratory refrigerators are used to cool samples or specimens for preservation. They include refrigeration units and freezers for storing blood, plasma, and other products such as virus specimens of CSF, outer ear, and unpreserved feces and urine, as well as vaccines and other medical supplies. This bench is our respiratory bench. Here we process aerosol transmissible disease contained in the Level 3 biosafety cabinet, including suspected AFB sputum and bronchoalveolar lavage samples. During the procedure, you are asked to cough deeply into the sterile container. Your provided sputum sample is smeared on a glass slide and look under the microscope for detection of Mycobacterium tuberculosis, which is a causative agent for TB infection. Almost all laboratory personnel handle water bath, which is another way to incubate at a constant temperature. This includes warming of reagents in the laboratory, incubation of cell cultures, and is preferred heat sources for heating flammable chemicals. A clinical lab test is also performed in the bacteriology section, which is to measure inflammation of your RBC. An indicative of inflammation is analyzed through an ESR analyzer. This measures the rate of inflamed red blood cells in whole blood that settle quickly relative to normal blood cells, revealing conditions such as anemia, renal failure, and obesity. Finally, when you get your blood drawn at the hospital, they must be separated, which is why the medtech must separate its components like the red blood cells, white blood cells, and platelets from the plasma for observation. The blood is placed in a tube inside of a centrifuge. As the blood spins, the heavier particles go to the bottom and the lighter particles go to the top.
So what is the importance of microbiology section in the hospital? The isolation and the characterization of microorganisms causing the infection performed by the microbiology laboratory. Microbiology laboratory has two important functions. The first is the clinical, everyday management of the infection. So the second one is the epidemiological. Knowledge on infected microbes in patient can lead to finding its source and route of transmission. This allows the staff to stop infection from spreading. Furthermore, the microbiology laboratory interprets the microbiology data for clinicians and physicians. The microbiology laboratory also participates in the development of an institution antibiotic policy. The microbiology laboratory is an essential part of an effective program and has many rules of outbreak management, performing the additional tests for epidemiologic studies, bacterial and fungal typing, surveillance, and reports about the new alert microbes of unusual resistance. During an outbreak or an endemic situation, when the causative agent is known, the microbiology laboratory is very important in diagnosis, as they may be able to use the selective media for the agent in question of minimize expense and increase the sensitivity of the culture.
뭐야 이거? 아이 괜찮아. Eventually,